150 million years ago, early dinosaurs and protobirds grew much like modern reptiles. Their mass increased in slow, intermittent stops and starts over several years. We see this in the limestone of the fossil record. Studies of Archaeopteryx suggests its growth was slow, mirroring the non-avian dinosaurs it lived alongside. But the modern lineage, the Neornithes, diverged from this pattern. They evolved to grow continuously and at much higher rates than their prehistoric ancestors. This shift shortened the time chicks spent vulnerable on the ground and accelerated the development of the musculoskeletal system required for flight, a transition that allowed birds to colonize nearly every niche on the planet. Today, all modern birds follow a sigmoidal growth curve. Growth begins with a steep period of post-hatch acceleration, reaches a peak, and then decelerates as the bird approaches its adult mass. To achieve this, species use different ecological strategies. Altricial birds, like starlings, hatch naked and helpless. They can't thermoregulate or find food, so they divert nearly every calorie provided by their parents into extremely rapid growth. In contrast, precocial birds, like quail, hatch fully feathered and mobile. They trade maximum growth speed for the ability to forage and flee from predators almost immediately. This table quantifies the difference. The starling's growth rate constant is 0.405, nearly 4 times faster than the independent Japanese quail, which sits at 0.106. Despite these different lifestyles, the underlying hormonal engine that dictates the shape of that growth curve is the same. To find the origin of these growth signals, we have to look past the feathers and bone to a small cluster of cells at the base of the brain, the pituitary gland. This is where the body synthesizes and secretes growth hormone, the primary messenger that initiates the post-hatch developmental sequence. The structure of this hormone is highly conserved. The amino acid sequence of growth hormone in a chicken is virtually identical to that of an ostrich or a penguin. This lack of variation suggests that the protein shape is so precisely matched to its receptor that any mutation would likely break the connection. Because the entire body depends on this signal to build new tissue, such a failure would prevent the bird from ever reaching a viable adult size. When the green growth hormone diamond reaches the liver, it docks with a matching receptor. This activates JAK2, which phosphorylates the purple STAT5 proteins, moving them into the nucleus to activate genes. This releases IGF-1, the hormone that performs the work of growth. IGF-1 travels through the blood to reach skeletal muscle and fat tissue. There, it forces satellite cells to proliferate and myoblasts to multiply, physically building the bird's body. However, this process requires active thyroid hormones, T3 and T4, to serve as synergists. Without a functional thyroid, the IGF-1 signal cannot be fully translated into new bone and muscle. Avian growth is not a single command, but a multi-step chemical cascade where a failure at any stage can halt the entire engine. We can see the consequences of a broken link in this chain by looking at the sex-linked dwarf chicken. These birds produce growth hormone normally, but a genetic defect in their receptor means the hormone cannot bind to the cell. The signal to grow is sent, but it is never received. This chart shows the result. In the dwarf chicken, represented by the pink bar, the lack of hormone binding causes a massive drop in IGF-1 and T3 levels. Compared to the normal control bird in blue, their total body weight is significantly reduced. A single microscopic error in a receptor shape determines the physical scale of the entire organism. Nature uses these same pathways to create the vast size differences we see within a single species. This is sexual dimorphism. Genetic dosing on the Z and W sex chromosomes can alter the baseline of these hormonal outputs between males and females. While male turkeys are programmed to be larger, the code is flipped in species like the Australian emu, where the female is the larger of the two sexes. Adult mass is a flexible genetic program, precisely calibrated to meet the needs of a bird's specific niche and environment. But even the most robust genetic program must contend with environmental reality. When a bird faces starvation or extreme stress, the body releases binding proteins that lock up IGF-1, preventing it from interacting with muscle cells. This actively stalls growth to preserve energy for basic survival. Humans have used these mechanics for our own ends. This data shows the impact of intense artificial selection. By providing unlimited nutrients and selecting for birds with hyperactive growth pathways, modern broiler chickens in green outgrow their unselected ancestors in purple by an order of magnitude. From the intermittent growth of Jurassic ancestors to the precision of modern poultry, the history of birds is recorded in their biochemistry, a system where every milligram of mass is the result of a specific, coordinated chain of events.